Hello and welcome. We're happy to have you with us this evening and to share this webinar, which we recorded earlier today. We hope you are excited as we are about the new cellular nutrition assay offered by Cell Science Systems. Our presenters are Roger Deutsch, founder and CEO of Cell Science Systems and lead developer of the ALCAT test and the cellular nutrition assay, and Amy Pazarka, director of nutrition services for Previmedica Group.
Okay, so what we're going to cover today is an introduction to the cellular nutrition assays that Cell Science Systems has to offer. And last year we completed a beta test, and we'll give the results of that beta test as well. So generally we want to cover the background and advantages of cellular testing versus serum testing for micronutrient insufficiencies. The dynamics surrounding symptoms related to immune responses and nutrient status. Explain our assays. There are three assays: the micronutrient assessment, the redox assay, and the antioxidant protection assay, and introduce you to how these tests are scored and how the information is reported. Before getting into that, I wanted to just give some context here, because we're facing now an unprecedented, at least in the last 100 years, a pandemic, which has to do with the immune system, because it's we find that more often than not, it's the immune system response that is causing fatality. It's very interesting. Just a few weeks ago in Circulation, and it was reviewed just last week in the New England Journal. A group from Germany investigated through autopsies of deceased patients what was going on physiologically, and they found that the inflammatory response to the COVID-19 virus was causing microvascular thrombi, and that this was present in tissues outside the lung, but also the kidney and the heart. And the characteristic of it, which will relate to our discussion here, is that they found that these microthrombi contained neutrophil extracellular traps associated with platelets. And that might be a new term for many people. Neutrophils, of course, are the most abundant granulocyte in our peripheral immune repertoire of immune cells. And sometimes neutrophils undergo a process called mitosis. And I want to talk about that a little bit, because that seems to be a significant factor in fatalities associated with COVID-19. The data that they acquired showed the distinct neutrophil and platelet activation states in COVID-19 patients. So immunothrombosis is not just an effect, it's actually a cause of the tissue damage and the organ failure and fatalities. And again, it's coming from an overactive immune response. However, when they stratified the population and they looked at, of course, the fatalities, they found that the immune system was hyperactive. Some people are referring to the immune system freaking out and there being a cytokine storm of some sort. There are different variations of cytokine storms. However, when they stratified and looked at the intermediate cases, and as we know, there are a number of cases where people have mild symptoms or even infected but are asymptomatic. But the intermediate symptoms where the immune system did not go into hyperdrive and cause a fatality, but caused serious illness, was due to a hypoactive neutrophil phenotype. So we have to look at this as a situation where the immune system is not underactive and not overactive. Now, one of the interesting things that came to my mind when I was reading that Circulation article was something I read earlier about how sterile inflammatory triggers, in this case, the researchers were looking, as they reported in Cell in 2018, a population of mice that was bred, that are bred to have display weakness in the cardiovascular system, and they're susceptible to cardiovascular disorders. And they found that underlying the pathology was a induced trained immunity affecting the neutrophils, induced by Western diet. So typically, experimental mice are given a mice diet, a chow diet, but they experimented with a mouse version of a Western diet, and they found out that the immune cells got into a hyperactive state and were trained and continued to stay in that hyperactive state, even after they switched the mice back. The immune cells would stay with a number of genes that promote inflammation were upregulated, and that persisted for quite a long time. So I think there might be a connection there between the kind of diet that people eat and the severity or the inability to cope with the COVID-19 infection. And of course, the people who are most susceptible are the ones who have underlying conditions, principally cardiovascular disease, diabetes, and obesity. And of course, all these conditions have as their common basis chronic inflammation. So I think we're connecting the dots here, and that the failure of so many people in the United States to survive infection, much more so than in other societies, is attributable to diet. So on the one hand, we need to find a way to achieve a balance in the immune system. This I just wanted to show for the people who may have not heard about NETosis yet. But this is actually a micrograph of a neutrophil that is undergoing NETosis, and the cell, even after it's dead, becomes about 16 times larger and throws out what are called neutrophil extracellular traps. Other granulocytes can do this as well, like eosinophils, and it's called ETs, and macrophages, and it's called METs. The interesting thing about these extracellular traps, what you see here is that the cell is breaking down and throwing out these extensions, which are comprised of the cell membrane, the membranes from the vacuoles inside the cells, the contents of those vacuoles, which are toxic mediators. That's what neutrophils do. They destroy pathogens. And also histones and other chromatin factors, including DNA. And they trap these pathogens, as we see here, and the DNA, which is very toxic, destroys the pathogens, hopefully. And then this mess is then digested by enzymes called DNases and RNases. And one hopes, if they're not destroyed, if their system is overwhelmed, if there's a defect in the enzyme system, the cell-free DNA that has emerged through these extracellular traps is toxic to the host, and there's a lot of bystander damage. And this is the kind of bystander damage that is being seen by the Sturman group in COVID-19 fatalities. So we have a direct link between overactivity of the innate immune system and fatalities from COVID-19. And that's what it looks like on a microscopic level. This was Scientific Picture of the Year taken by Dr. Volker Brinkmann at Max Planck Institute. It was the picture of the year 2004. And in 2004 is when the literature began to see articles about NETosis at a very small level, and now it's becoming much more popular, and there's a lot more research in it. In fact, Cell Science Systems is currently engaged in an international research project looking at NETosis and cell-free DNA following immune cell challenge with foods. And I think we're going to have some very interesting results from that study that we'll be presenting and will be published fairly soon. Now, again, before we get back into the particulars of our cellular micronutrient assay, I just want to spend another few moments on the other part of the immune system, which I think we have to address if we want to achieve the immune balance, which is desirable for the body to be able to resist infection, maintain homeostasis, but not have the effects of the immune system's overactivity harm us, even to the point of what we see with COVID-19. So I want to speak about food sensitivity. The innate immune system, which we're discussing so far, is what is associated with food sensitivity. Classical allergies are associated with the specific immune system. And for now, if you could just hold that distinction in your mind, we'll get back into, in just a moment, explaining the micronutrient assays, which address improvement of specific immune function. But of course, the adaptive immune system, or the specific immune system, and the innate immune system, the granulocytes and the dendritic cells that present allergens to the specific immune cells, they work in harmony with one another. So it's important to address both of those. And of course, the biggest factors that underlie healthy immune function are, number one, having proper nutrients that are required for immune function, for cell growth. Number two, having the ability to not overreact to danger signals, but be able to respond appropriately and effectively. So we highly recommend, when looking at nutritional assessment of a patient, that one also assesses the diet from the perspective of whether the diet is compatible for that person. And for a number of years, we've developed and have been offering a cellular approach for doing just that. That's most of you have heard of if you're not already using it, which is called the ALCAT test. And briefly, how that works is that we want to measure the size of the cells as they are exposed to an allergen, because we know from looking at the previous micrograph that there are certain stages a cell goes through when there's it recognizes something as a threat. So the cells actually become larger and will extrude contents from its vacuoles internally as well as externally, and then ultimately may extend into the process of mitosis. And the way to determine that, which is independent of any particular biological mechanism, is to look at the reaction of the cell when it's exposed to the substance while the cells are still viable. And we use this method whereby the whole blood of the test subject is incubated with buffers, natural buffers and so forth, and an electrolytic solution, and is aspirated through an electromagnetic field that will show a change in impedance proportionate to the size of the cellular particle. And from that, we're able to generate what is called a histogram. And this would be a depiction of what happens in real time as the cells flow through that kind of a channel and there is a reaction. So let me explain. Here on our histogram, first of all, the red line is the patient's sample without any substance added to it. And a number of these controls are run to be averaged and form a baseline. And then individual test samples exposed to individual test substances are also analyzed one by one. And one by one, they are plotted against the baseline. In this area of the graph are the lymphocytes. And these are the cells that are the cells of the specific immune system. And again, just hold that in mind. I want to talk a bit more about the cells of the specific immune system in the context of our micronutrient assay. These, of course, are the granulocytes, or the cells of the innate immune system. And this is typical of a kind of a reaction where we see a lower cell count because cells have been destroyed, and some of them are still in the process of becoming larger. This y-axis is size. As you move to this direction, they are bigger. So some of these cells got larger, and some of them actually fully degranulated. And maybe if time were allowing, they would progress into a process of mitosis that we have seen. So now I want to begin the discussion about specifically how the cell science systems nutritional assay works. The original concept came from a study back in 1941 from researchers at University of Chicago. It's a very famous study because it demonstrated the role of genes and enzymes. At first and this has become, as most of you know, the DNA dogma: one gene, one enzyme. Later revised to say one gene, one polypeptide, because not all proteins, peptides and proteins that are manufactured based on the genetic expression are enzymes. So their study was very interesting. The thing that came out of it that's of relevance to our discussion here is that when altering the cell culture medium, you impact on the growth of the cells in the medium. In their experiment, they took a common bread mold called Neurospora casa, which they were able to grow in an essential growth medium, a minimum culture media. They then irradiated the spores and found that the effect of the radiation was such that the spores were no longer able to grow in a basic medium. So obviously they had deranged some genes, and the genes then were not functioning, the enzymes were not functioning in such fashion as is required to support the growth of the spores. So they looked at adding back, they divide the radiated sample with unradiated spores, and they would add back various categories of nutrients, vitamins, aminos. Then they would, of course, continue with the minimum growth medium as a control and a complete medium. And they found out through this trial and error that when they got to testing arginine, they were able to restore the growth of the cells. Similarly, one cause of micronutrient deficiencies can be an inborn error of metabolism. Here they induced an error in metabolism by damaging genetic material intentionally, but it can happen certainly naturally that a person's enzyme systems do not all function properly, and there might be a need for additional micronutrients to support those pathways to support growth. Now, as mentioned already, and this would be no surprise, the immune system requires, again, to function when it needs to function, and then the other half of the immune system will come in afterwards and repair the peripheral damage that has been wrought by the attack of the immune system against a threat. And they must work in that kind of balance. So half the immune system goes out and kills and is very aggressive, and that's sort of a Yang principle. This, by the way, actually is the cover of Science magazine, January 2013 edition. where, by the way, if you can get your hands on it, is a great special issue about immune function. And the overarching theme is that we must have balance. Now, to look directly at the specific immune system, I want to give a little bit of understanding of the basic function of how specific immunity works. So unlike the innate immune cells, which recognize, that have pattern recognition receptors that respond to motifs that are found on the cell surfaces of bacteria and fungi, and pretty much all of the granulocytes will recognize it. When there's a failure or an inability of the innate immune system to completely quell an onslaught from a dangerous pathogen, the specific immune system comes into play. So why would that happen? Of course, the way the immune system, the innate immune cells would destroy a pathogen is that they would take it up, traffic it to internal lysosomes. There would be a number of enzymatic activities that would cause generation of free radicals and various toxic mediators that would destroy it. But if the pathogen, for example, has a certain kind of coating, polysaccharide coating that hides it or makes it difficult for the cells to grab that pathogen, they might try this trap, external trap pathway, or they might generate or activate complement proteins, which will coat or opsonize the bacteria. And if that fails, then we need a specific antibody that will bind the pathogen and enable it to dock onto the phagocyte so it can be destroyed. So where do those antibiotics come from? They come from the specific immune system. So that's why this is theoretically a more evolved, more specialized branch of the immune system, and it behaves entirely differently. First of all, for the specific immune cells to become activated, the antigen or the pathogen, which serves as an antigen, is taken up by an antigen-presenting cell. It's broken down. Its peptides bind with a molecule, a transporter molecule called a major histocompatibility 2 molecule, which then transports to the surface where it will be recognized. And antigen recognition is actually the key here. Only certain lymphocytes, T lymphocytes, will recognize that pathogen. Only the ones that have receptors that conform to the motif of that pathogen will recognize it. And that recognition is critical in order for the T cells to become activated. And in fact, there's even a secondary signal, which if that doesn't engage, it's not depicted here, but there is one. And if that doesn't engage, the T cells do not become active against that pathogen. They can actually become anergic and non-reactive and ignore it. And in fact, that's how the body develops tolerance during early days of exposure. What happens then? So there's very few cells that recognize a particular pathogen. Those cells may be naive T cells that then go on to clone themselves, or there may be cells that saw that pathogen before. And that's the entire concept behind vaccination. The idea of vaccination is to give a little bit, maybe an attenuated form of a virus that is sufficient to activate T cells so that they recognize it. And then the next time they see it, they can mount a more robust response more quickly. What does that mean? There's few cells that recognize it. So these cells, unlike the innate cells, will then go on to divide. So under the right circumstances, lymphocytes move from a resting phase to an active phase, and then they divide. Now, here's the key point. This division of the cells, of course, is necessary in order to move on and send the signals to B lymphocytes to produce antibodies, or to develop into cytotoxic T lymphocytes that will destroy other infected cells or cancerous cells. But the key here is that they have to divide, because there's only a few memory cells or a few naive cells that will recognize the pathogen in the first place. The division of the cells that takes place is driven, of course, by signals that occur, but they require nutrients. So the basis of our micronutrient assessment is to look at the rate of cell growth when we add specific micronutrients into the culture. And you can easily from this understand the rationale behind that. Now, just to put into perspective how important these cells are, in the National Health and Nutrition Examination Survey that went on for many years, 31,000 plus participants were looked at, and they stratified the populations according to many different factors like inflammation markers, CRP, and hematologic factors, RDW, but primarily lymphocytes. This is the title of the paper that came out in JAMA late last year. Immunology Association of Lymphopenia. Now, penia, that suffix means a lower count, so a low lymphocyte count, association of low lymphocyte count with risk of mortality among adults in the general population. So they found that patients in this long-term observational study who had a low lymphocyte count had a much higher death rate, a much higher mortality risk. And if you added to that other hematologic abnormalities, and particularly inflammation, that accentuated it. And you can look at the data later on. We don't want to get too sidetracked, because we want to get back to our main focus, but the change in mortality rates was very, very significant. So we need those cells to function properly. So having the ability for those cells to grow and proliferate is something that represents a valid marker for immune health. People, we wanted to also weave in, so many people were asked to provide their questions. We had several questions about genetic testing for micronutrient deficiencies. Now, at Cell Science, we do some genetic testing for particular micronutrient deficiencies, but we focus on genes where it's really well known that they play a role, because we're still, even though the Human Genome Project was completed about 18 years ago, we still don't have enough information that just by genetic evaluation alone, we can diagnose disease, much less identify nutritional deficiencies. We have a few instances like methylene tetrahydrofolate reduction. If there are defects in that particular enzyme and other enzymes in that pathway, there will be too much homocysteine, and that is very toxic, and it's fairly clear that's a major risk factor for Alzheimer's and cardiovascular disease. But that fact's been known since the 60s, thanks to Kilmer-McCulley, who developed the homocysteine hypothesis. But beyond that, information is becoming available as studies evolve. But when asked about whether the genome, play on word, has a genogram granted our wish yet. The editor of the New England Journal, in an editorial with David Hunter early last year, said that, well, we're not there yet. The genome has not yet granted our wish to tell us everything we need to know. And the reason is that there are very few monogenetic diseases. Most of them involve thousands of gene variants, and some of them have a small contributory effect, and you need to go through an iterative process with thousands of genes to try and associate that with disease conditions, and we're not there yet. The other factor is that we have epigenetics. And epigenetics, a clear understanding of that is that it's gene expression. And there are epigenetic modifications we've all heard about, methylation, which happens at CPG islands, which in the promoter regions will suppress gene expression. And of course, in embryonic development, we go from 1 cell to 50 to 60 trillion cells, and cells differentiate by turning on genes that are needed, but turning off the majority of genes when they're no longer needed or they're just not required. Despite the fact that every cell has the entire genome, different cells obviously express and require different genes to be active for their products to enable them to perform their function, but the rest are science. Other modifications, like histone acetylation, causes changes in the nucleosome, which will cause the expression of certain genes. And these things are all impacted by everything we do, think, smell, eat, move, don't move, and so on and so forth. And clearly, when we see that two people with the same identical genome can have different phenotypes depending upon their epigenetics, their lifestyle, we see how significant the effects of epigenetics are. So there's no doubt that in the future, this will evolve more and more. There are new technologies coming about that enable us to examine the epigenome more accurately, and that's happening very, very rapidly, and there's a lot of progress there. But there's still a lot of work that needs to be done. So we should stay at it, but in my opinion, given these facts, I don't think we can, except in a few instances, look at the genome and determine what the diet ought to be. So if that's confusing to you, don't feel bad. A nice article, there's a lot of these interesting articles coming out about trying to explain to everybody what all these terms that we hear, protective antibodies, herd immunity, CD8 positive T cells, which kill infected cells, interleukin-1 beta, interferon gamma, which are factors in cytokine storm. Although there are also others, but type 1 interferons play a big role. Interleukin-1b is a major factor. But the title of this very interesting article in Atlantic was Immunology is Where Intuition Goes to Die. So what we think makes sense may not be how the body has a different way of doing stuff than what our intuition may think it ought to do. And that's too bad, because we really need to understand how the immune system reacts to the coronavirus. And again, I connect this with that cover of Science magazine, where, in order to really understand the immune system, you almost have to understand the Tao Te Ching and the yin and yang of life. And that's just the way it is. Some of the folks who were the pioneers of quantum mechanics would scratch their heads at all the crazy findings that they found in the physical world. And some of them went off and studied Oriental philosophy in order to try and make sense of all this. So we're faced with this kind of situation where we have a system, the immune system, which is super complex. And we understand it piece by piece, and then sometimes we find out that there's more to it We used to think that the immune system was just there to distinguish between self and non-self, but that's not the case. One important investigator said, no, it distinguishes between danger and non-danger. But then sometimes it doesn't do that. Sometimes it thinks something that's non-dangerous is dangerous and overreacts and have an altered reaction or an allergy. And increasingly, we have situations where the immune system is turning against the host and we have autoimmunity. And these things obviously aren't right. It's a complex system. It's more like a Ferrari than a 57 Chevy. It's not so easy to work on. There's a lot of moving parts. There's a lot of sophisticated technology in there. And an analogy like that does no justice to it. Plain and simple, the immune system is complicated. So looking at functionality of the immune system just seems eminently logical. So that's basically what we do with our cellular micronutrient assays and the variations of that, which are the antioxidant protection assay and the redox assay. So summarizing, poor nutrition compromises immune function. The cells need to become metabolically active. When there's a threat, the resting cells become activated. That consumes nutrients. So intracellular nutrient status is crucial for adequate functioning of the immune system. Absolutely. Nutrient supply is inadequate. The immune system is deprived of the components needed to generate an effective response. If there's an infection, one of the defenses is to kill it by generating free radicals. So we need good nutrition also to protect the immune cells themselves and tissue cells from the side effects of the generation of reactive oxygen species and reactive nitrogen species that are aimed at killing danger molecules. So immune function may be improved by restoring deficient micronutrients. So what's the best way to do it? So we can look at the serum and say, there's enough vitamin B6 according to statistical requirements, because we tested X number of subjects and we generated a distribution curve. And if you look at the bell curve, that should be sufficient. Well, those statistical models don't work. The man who coined the term biochemical individuality, William Rogers, wrote that book, Biochemical Individuality, in 1956. After he himself, when it was admitted to the hospital and the night before surgery, was given morphine to put him to sleep, and it kept him up. So these paradoxical reactions, although they're not that common, occur. And it gave him pause to think, like, why am I having that effect and why not others? And he made an extensive research and found out that, hey, we're all different. We share a lot of the common genome, but a little bit of variation may account for huge differences. So looking at statistical models to try and determine whether or not a particular micronutrient is sufficient for a particular individual may or may not work. Certainly it has some value if it's way out of range, but that particular individual may need more for a number of different reasons. An inborn error of metabolism, for one thing, other stress, infection, trauma, pregnancy. So let me turn this over to our other presenter, Amy Pizarka, who will explain a bit more of the details of our testing.
Thank you, Roger. It was a fascinating presentation regarding the immune system. So with regard to the differences of the tests that are available to assess micronutrient status, we do get this question quite a bit. We got this pre-webinar as well. What are the differences and what makes ours unique? Well, the biggest question being the differences with the measurement of the micronutrients in the plasma and the serum. So keep in mind, looking at micronutrients in the plasma or a serum, it's a reflection of their levels in circulation outside of cells. extracellular. It's not a reflection of the nutrient levels within the cells where the nutrients do their jobs and where metabolism happens. It's also not an indication of the nutrients' functional impact on the cells, as Roger described. So the other thing to keep in mind is the serum plasma levels of the nutrients are prone to daily fluctuations, and they're considered more of a snapshot. So what the nutrient level is at that one time when the blood was drawn. And it's really a reflection of the patient's most recent consumption of those particular nutrients. And it's very possible for nutrients to fall within normal limits in the blood and the serum and the plasma, but be insufficient in the intracellular tests, and vice versa. Other questions about other tests. This question came in, the red blood cell analysis. That actually could be advantageous over serum and plasma levels. So there are advantages to all these tests, really. But keep in mind that with the red blood cell analysis, good for certain nutrients, however, not providing insight into any functional response of the micronutrients on the cells. And you might take a look at metabolites in urine to assess nutrient status. Well, that would reflect the nutrient status within a 24-hour period for that patient, and also rely on how concentrated the urine is. So, considering all this, assessing micronutrients at the cellular level and their functional impact on the cells is preferred. It provides insight into an individual's long-term nutrient status over about 6 months versus the short-term variability that you'll see with these other tests. So one way to think about this, and you might have thought of this, of course, on your own or read about this, but think of the hemoglobin A1C test that we're all used to looking at, the long-term test for blood sugar, blood glucose control versus a snapshot taking the fasting blood sugar. So it's kind of the same thing. So the first assay is the cellular micronutrient assay. And as you know, micronutrients are required for every metabolic process in the body and necessary to produce energy and strong immune system, of course, as Roger described, synthesis of enzymes and hormones and neurotransmitters, and production of tissues and organs and bones. And they protect the body from oxidative damage. Disruption in cell metabolism, which of course happens when we have insufficiency of these nutrients, can certainly impact overall health and lead to chronic disease. This is showing you, which is a question that comes in quite often, is what nutrients are tested in the CNA. So you see all these nutrients listed here, and there are 52 nutrients tested. So this slide is to show you that metabolism happens, and to remind you that metabolism happens and occurs within the cells. All metabolic and biochemical processes take place within cells. And as Roger discussed, discussing the specific and the innate immune systems, with regard to the when the body has an infection, the body is increasing the production of the T and the B lymphocytes, and they recognize and combat the specific invader. And the faster that the cells can reproduce, the faster that they can overcome the infection. So again, as Roger mentioned, the ability of the cells to multiply is driven by our nutrient stores, and many factors impacting nutrient requirements. So they're unique to all of us. Our genes, as Roger mentioned, stress, how much energy we're expending with activity and exercise, pregnancy, being sick, having an infection, toxic burden, our sleep patterns, all of these things impact our nutrient demand. And as you always hear about nutrition, one size definitely does not fit all. So assessing and monitoring your patient's individual micronutrient needs at the cellular level does enable you to use safe and effective, individualized and truly targeted nutrition strategy. So that with regard to the testing process, Roger will discuss that and describe that to you.
So obviously many people are familiar with using other micronutrient functional tests that involve lymphocytes. And I can tell you many people have asked, how does our test differ? So I'm being very familiar with other methods of measuring lymphocyte proliferation. We've developed a few of those. And the way our test works is that we separate the lymphocytes from the whole blood using a density gradient and centrifugation, which is a pretty standard approach. And what we do that's different at that stage is that we add back the patient's serum to the lymphocytes. This probably is the most innovative difference between the cell science, our test, and the SpectraCell test, which many people have been accustomed to using and are familiar with. I'm pretty familiar. I've had a lot of interaction and some collaboration with the inventor of the SpectraCell test, and when their former scientific director from their early days. And what essentially they addressed in their technology was the licensing of a culture medium that was developed by Dr. Shah from the University of Texas in Austin, which enabled them to have a consistent medium without the addition of fetal calf serum. Because fetal calf serum could, biological could vary from lot to lot. So what Professor Shah developed was an essential growth medium that was serum-free, which can be manipulated by adding or deleting micronutrients. And the concept's great, but I'm not sure it really worked. I think he did a fantastic job, but even he told me that he wasn't happy with the test and encouraged me to develop a test many years ago. It was an heroic effort, and I think there's a tremendous amount of value in that. But without serum, it seems like it's a bridge too far. And that's why sometimes there's a lot of, often there's rejections. So it's a simple and common sense answer. We developed the technique by adding the patient's own serum, which provides sustenance to the lymphocytes. And of course, that is the in vivo situation. The patient cells are in the patient's serum. So why not use the patient's serum rather than foreign serum? We stimulate the cells with the mitogen, and we measure the baseline proliferation rate by using, this is another difference, instead of using thymidine, triturated thymidine pulsed into the cells after a period of incubation, and then extracting the DNA and counting the amount of thymidine that's been incorporated into the DNA with a special device, a beta scintillation counter. We don't feel that the use of radiation is necessary or desirable. And in fact, in the literature, you'll find that there are a number of methods that can measure lymphocyte proliferation without using radioactivity, but rather measure the rate of metabolic activity of the cells, which does correlate with the proliferation of the cells, as well as does DNA synthesis. So to just recap, we separate the lymphocytes, so those are the cells we're interested in. We add back the patient's serum, we separate the sample into a number of different aliquots, We stimulate all of them with the mitogen, but in individual test wells, we add the individual micronutrients. And then after a period of incubation, we look at the differences of the metabolic activity of the lymphocytes in each individual test well. And here you can see the only variable is the presence of a specific micronutrient. So the question, the research question is that, does that micronutrient enhance this desirable response of increased metabolic activity in the lymphocytes, or does it not? And to what degree? It's quantitative. And if the specific micronutrient is found to significantly enhance the metabolic activity of the lymphocytes when stimulated by a mitogen. And again, a mitogen acts like a pathogen, except it's more universal. It means mitosis generator. And we just run our calculations, and when we find that the micronutrient has enhanced proliferation, then we report that out as something that is insufficient. And then we have a gradient which depicts the quantity of that response. Amy?
All right. So here's an example of part of the cellular micronutrient assay test results. Of course, as you know, there are many more nutrients than this tested, but we enlarged it so that you could see how it's reported. So as Roger said, the nutrients that enhance the functional response of the cells, in other words, the proliferation rate and the metabolic activity of the cells are increasing over baseline, they're going to be reported as borderline insufficient or insufficient, depending on the degree of the increase in the activity of the cells. So if you see take a look at the gradient score, the results that are reported in the green to the far left, or where it says no proliferation, or the notches are within that green area, that represents the patient's baseline proliferation rate. So in other words, the nutrient did not increase the metabolic activity of the cells. When it's increased slightly in the yellow area, you'll see them reported there as a borderline insufficient. And when the activity of the cells is increased significantly, you will see them reported in the red area, and the nutrients will be reported as insufficient. So when the nutrients increase the metabolic activity of the cells, that is a good response, of course, but it's also an indication that there's insufficient amount of that nutrient in the original specimen. Otherwise, the activity of the cells would not have increased with the addition of that nutrient. So this particular patient's test results indicate biotin is insufficient, riboflavin, vitamin B6, vitamin C, and vitamin K. And then with regard to borderline insufficiency, it's also important to take a look at the folate, vitamin D, and I believe selenium, but you can't see that here. So when I look at this, of course, this is helpful for practitioners to connect the dots and put pieces of the patient's information together to kind of determine what is going on. But I would wonder about methylation. I would wonder about the patient's homocysteine level when I see riboflavin and folate and B6. Crucial for balanced methylation. When I see those are insufficient, it makes me wonder. I wonder how he's doing with his homocysteine levels, but also wonder if there's an MTHFR variant that he might have, which would impact his the ability for the active form, the conversion of to the active form of the folate. So these are all things that we can correlate when looking at test results. So it's very, very helpful to be able to connect the dots and put the pieces of the patient's information together in order to come up with an individualized nutrition intervention. Now you might notice that we're not calling it nutrient deficiencies. And it's best to think of these as subclinical deficiencies or metabolic deficiencies, and you might expect your patients to be exhibiting signs of deficiencies by noticing that they're insufficient in particular nutrients, but you're likely not seeing symptoms of overt deficiencies. And for example, vitamin C here. More than likely, a patient is not exhibiting signs of scurvy. Now, certainly if we don't address this, we don't do anything about it, we don't restore and replete, certainly the patient could have a problem later on with overt deficiencies, but especially, if we don't address the problem, we're going to be dealing with another chronic disease. So important to keep in mind. So with regard to descriptions, within all the test results in the micronutrient assay, for the nutrients that are considered insufficient or borderline insufficient, there will be a description of each nutrient included in the test results. And the description will include what the nutrient is important for, the most important functions, and what the nutrient may be useful for the prevention or treatment of, and good food sources of that particular nutrient. So we encourage you to take a look at that. And there's a booklet that comes along with this test called Understanding Your Cellular Nutrition Assay. And in that booklet, every nutrient, every botanical, and every antioxidant nutrient, everything is described in that booklet as well. So important points to keep in mind with regard to these nutrient insufficiencies. It's important to figure out what is contributing to the nutrient insufficiency. So these tests are best used within the context of a comprehensive assessment in order to connect those dots. So we need to consider medical conditions, your patient's reported symptoms. We need to assess gut health. We need to look at food sensitivities, the genetic test results, other lab data, prescription and over-the-counter medications. All of these things are to be considered, and obviously the patient's eating pattern and the patient's supplement routine. We all know this, but your patients might ask you, how could we possibly have insufficiencies in nutrients? Because we have such an abundant food supply in our country. And of course that's true, but we all know the standard American diet is abundant, but unfortunately abundant in highly processed packaged foods that are loaded with calories and not good sources of nutrients. So many of our patients are not meeting their nutrient needs. They're not consuming the amount of plant-based foods, fruits and vegetables that we would be recommending. And even if your patient reports to you that they're consuming a health-promoting eating pattern, it's important to realize that we just can't assume that they're meeting their nutrient needs. Many reasons, but one, we know modern farming has led to soil that is depleted, which is leading to depleted crops, nutrient-depleted crops. So once again, we cannot assume our patients are nourishing their bodies, nourishing their cells properly. But what about other reasons behind insufficiencies? Keep in mind that the cellular nutrition assay, the cellular micronutrient assay, identifies the insufficiencies. It's up to us as practitioners to help the patient determine what is leading to these insufficiencies. So before we can come up with an intervention that will be helpful. And a lot of things need to happen for a nutrient to function optimally. Obviously, we need to consume it first. Then these nutrients need to be absorbed into the gut, released into the bloodstream, taken to the cells of the body. The cells taking up those nutrients requires membrane transport systems, receptors and enzymes and helpers, cofactors in the process. So the point is there are opportunities for things to be inefficient, leading to these insufficiencies. So that's important to keep in mind. So these are some potential reasons, and hardly ever is it ever one thing contributing. But the first thing obviously would be the most basic thing to look at first is the patient's intake, their regular food intake and the supplement routine. it's a good idea not to just accept what the patient says regarding, yep, I'm eating pretty healthy. Really interview the patient to determine, well, what does that mean? Maybe if they're eating the same foods every single day, that's not a healthy eating pattern. So is the intake inadequate? With regard to supplementation, take a look at the supplements that, ask your patient to bring the supplements in, or ask specifically what brands they're taking. Might not be taking good quality supplements, might not even be getting the nutrients that they think they are. You, for example, have a patient who says they're a vegan. Maybe this is why B12 is insufficient, or iron, or calcium and vitamin D. Maybe they're vegans, not eating animal sources, but maybe they're not doing it correctly. Other things, we hear a lot, oh, I eat plenty of fruits and vegetables. But what if the patient's eating the same fruit every single day, the same 3 vegetables every single week, not consuming that variety? That could be something that's so basic to help your patient improve. Medications. We all know that, every medication impacts nutrient status somehow. So it's important to consider. Antacids, we know the problems with long-term antacid use. Maybe this is why B12 or iron or zinc is insufficient. Maybe this is also why maybe some amino acids are insufficient. Maybe your patient has too little HCl production in the stomach. Metformin, another medication that we're very familiar with for people with diabetes or pre-diabetes, impacting folate status, impacting B12. So all these things need to be considered. Unfortunately, fortunately, I guess, as nutritionists, not able to do anything about medication except hope that the improvement of the lifestyle and the nutrient status, correcting those nutrient insufficiencies as well, cleaning up the gut, addressing food sensitivities. The hope would be that the patient would not need the amount of medication they're taking, or maybe it even could be discontinued by the physician. So looking at gut health, looking at food sensitivities, looking at digestion, HCl production, of course the microbiome. Maybe we need to address the friendly bacteria population in the gut. Maybe there's dysbiosis going on. Of course, chronic disease, stress. Is your patient a drinker? Drinking too much alcohol, maybe impacting the B vitamin status, smoking, impacting the antioxidant capacity. All of these things impact or can be leading to nutrient insufficiencies. And as Roger mentioned too, genetic variants. So can definitely be impacting the nutrient availability within the cells, or again, probably a combination of these factors. So considering this, it's very important that patients to receive guidance from you, the practitioner, to know how to replete these nutrients. It's best, we really prefer that patients do not look at these results and not review them with you and say, oh, okay, I need B12, let me head to the corner drugstore or to the dollar store and let me get some B12, some cyanocobalamin, and let me be done with it. That's not going to be helpful at all. So your patients will be much more successful at improving their nutrient status by working with a practitioner such as yourselves, who can answer questions like this and proceed with an individualized nutrition plan. I just included this slide to remind you, once again, Roger mentioned methylation. And when you take a look at, this is an example of the front page of the Cell Science Systems methylation detoxification profile test that's offered. So these 5 genes here are representing the genes that are encoding for enzymes, of course. So, and the color coding results would be indicating if they're heterozygous positive, homozygous positive, or homozygous negative. So just wanted to remind you that this is one of the one of the ways that folate can be impacted. So this MTHFR variant here, this represents that this patient inherited a variant from both parents. So he's homozygous positive. So if we know that this MTHFR enzyme is very important for the conversion of the inactive form of folate to the active form of folate, which is the 5-MTHF here that we see in this green outline box. With variants here, with the enzyme activity lessened and not really getting maybe a good nutrient support here, that could certainly be a reason why the folate is insufficient. So it's important to be able to take a look at genetic test results whenever you have them, and we encourage you, of course, to combine the cellular nutrition assay with the methylation, the methyl detox profile that's offered by Cell Science Systems. With regard to repleting nutrients, we do get this question, and a few practitioners submitted this question prior to the webinar, asking if we will be giving guidelines for repleting nutrients. So, as I mentioned earlier, we encourage you to take a look at the descriptions of all the nutrients that are included in the test results, but also in the booklet, Understanding Your Cellular Nutrition Assay. But keep in mind that all of the factors that could be potentially leading to these insufficiencies need to be determined first before you know what intervention, what do you do, before you know if you should increase the patient's supplement if they're already taking it. So we need to consider all these things. Of course, that's the first step. But when you are repleting, of course, we would suggest that you're starting with food first. So you're encouraging the patient to meet their nutrient requirements as much as possible with a health promoting eating pattern, with plenty of plant-based foods and fruits and vegetables and so forth, unpackaged, unprocessed foods. Address gut health, address food sensitivities, very, very important. With regard to supplementation, if your patient's already taking a supplement, the question would be, well, here's an example. We have maybe, you might get a comment like this: how could I possibly have an insufficiency in folate? I've been supplementing with folate. All right, my question would be, all right, let's see that. What is the form of folate you've been supplementing with? We look at the form. How long have you been taking this? Very often you'll hear 2 weeks. All right, well, remember, this is a test that is a reflection of the patient's status, nutrient status, over 6 months. So that 2 weeks of that supplementing of the folate, you might just suggest that the person continue with that, but there's the reason why that they're still insufficient, because we're only looking at 2 weeks of that. So all these things need to be considered. With regard to doses of particular nutrients to suggest within supplementation, that is not something that's provided by us right now. We certainly will, like we do with all of the Cell Science Systems tests that are offered. We offer tutorials that go along with those tests, and they are all kept in the Cell Science Systems practitioner portal in your resource section, and we will continue to develop tutorials for this test as well. One of the things I wanted to remind you of, though, are these wonderful resources, though. We turn to these quite often. These are excellent resources that you could take a look at, talking a lot about the nutrients, what they're used for, the mechanism of action, doses that have been used in research. So I encourage you to tap into these. The other thing that I have not listed on this slide is doing a drug-nutrient interaction check. Very important also with your patients who are taking medications. Drug-nutrient interaction checker. The Natural Medicines Comprehensive Database, which I have listed here, the last one on the slide, that has a drug-nutrient checker. And another great resource is Integrative Therapeutics has a great one. It's called Integrative Pro. Their website is integrativepro.com. You go to resources, and then they have a drug-nutrient interaction checker there as well. So I would encourage you to utilize those resources. With regard to free radicals, so we all know free radicals are coming from foods and infection and smoking and so forth. All the factors that you see listed here, with neutrophil activation being the biggest contributor. And oxidative stress occurs when there's an imbalance between the formation and the removal of free radicals due to the overproduction of the free radicals and or impaired ability to neutralize. neutralize those free radicals and repair the damage that they create. So that's oxidative stress and, of course, oxidative damage. So that brings us to the redox assay. And the redox assay is a measurement of the cell's resistance to oxidative stress and its overall antioxidant capacity. In other words, the cell's ability to protect themselves from oxidative damage without the addition of any of the antioxidant nutrients. So their cells as they are. So what happens with this test is, once again, looking at the T and the B lymphocytes. Now at this test though, the cells are challenged with hydrogen peroxide to create free radicals. So the cells are essentially put into the state of oxidative stress. And with that happening, now it would tend to reduce the growth rate, or reduce the proliferation rate, of course, and damage the cells to the point of cell death. And that's what you would expect, but of course that's what we don't want. We want the cells to be protecting themselves. So the proliferation rates of the patient's cells, they're assessed after the addition of the hydrogen peroxide. Now for this particular result, you see that it indicates a cell loss because it was a decrease to 77%. The white diamonds you see here in the bar in the graph next to it, within the light blue area, that represents the patient's proliferation rate. So, and then they plotted it and compared it to the general population. Red being below average for the population, light blue being average and normal, and then of course the ideal would be above average. So of course what we say is, if they're in the red, of course, or in the light blue, let's say, how can we get this better? And if they're in the dark blue, how can we keep it that way? So that brings us to the antioxidant protection assay. So here are all the antioxidant nutrients and botanicals and phytonutrients that are tested in this assay. There are 48 of them here. And in order to improve the cell's antioxidant capacity, this APA assesses the benefit of antioxidant nutrients and botanicals, phytonutrients, anti-inflammatories specific to each patient. So there's really no need for us as practitioners to suggest antioxidants and just hope or assume that they're doing what they want them to do. I can remember, you might have this situation where you have a patient, who are health conscious patients, thankfully, but they're looking all over the internet and getting their nutrition information from the internet, and they're taking, a boatload of supplements, and they could come to you with a whole duffel bag worth of supplements and many antioxidants, of course. So actually, when you're seeing specifically which antioxidants are going to be protective, you might end up saving your patient money. So here's what happens with the APA and the testing process. This is how the test works. The cells are in the state of oxidative stress with the addition of hydrogen peroxide as they were in the redox assay. But now, in individual test wells of all these cells that are in oxidative stress, now antioxidant nutrients and botanicals and so forth are added one at a time. And then the proliferation rate of the cells is assessed after the addition of the antioxidant. So just like in the CMA, where we saw the gradient score, and you see you remember the green area to the far left represented the patient's baseline proliferation area. Same thing here in the light blue. To the left represents the baseline proliferation area. The light green represents a slight increase in the proliferation rate, so that's why these cells are now, the nutrients are called protective, because they're increasing the proliferation rate even while the cells are in oxidative stress, so they're protecting them from those cells dying. And then, of course, increasing the proliferation rate significantly in the dark green area. So you might say, should the patient take everything that comes out to be protective or highly protective? No, but we encourage you to take a look and decide which ones you would choose. My choice first would be going to the ones that are most highly protective. And the ones that came out to be insufficient in the CMA and also protective here, of course, with regard to the antioxidants I'm choosing, I would choose those first. For example, if you have delta tocotrienol that comes out to be insufficient in the first test and it's protective here, certainly that's one of the antioxidants that I would suggest. But the other thing to keep in mind here is a lot of these botanicals that are tested in this test are also tested in the ALCAT test for sensitivities to them. So another question that came in pre-webinar was, can we do this test along with the ALCAT test? And the answer is yes, absolutely, we encourage it. When you're doing the ALCAT test and you're going to do this test as well, we encourage you to test 250 foods and the 50 functional foods and medicinal herbs, because in the antioxidant protection assay, a lot of the botanicals that are tested here are also tested in that panel, the medicinal herb panel. So what I'd be looking at, for example, before I would suggest echinacea for someone who comes out to have echinacea protective, if they had the ALCAT test, I'd be looking at the ALCAT test to determine what the reactivity was there. If non-reactive, then I would feel comfortable suggesting it. If reactive in the ALCAT test, which could happen, remember they're testing different cells, I would say for the time being, while you're following your ALCAT test results, your ALCAT test with regard to echinacea would take priority following that before you would start using echinacea as an antioxidant for the time being. So again, we encourage you to take a look at all of these, to look at the test, to combine the cellular nutrient assay along with the methyl detox profile, along with the ALCAT test.
Thank you, Amy. Just to conclude, I think there was one more quick question. Someone asked, why do this test or any test if you just give a patient a good multi? And the answer that comes out is that it's better to be more targeted, and maybe you can modulate and give more of some, less of some, and none of some based on laboratory findings. But just to conclude now, many of you probably even participated in our beta study that we concluded last year, and there were several hundred patients where we looked for correlation between food sensitivity and lifestyle factors like drinking and smoking, dietary habits, and GI conditions. And it's been published now, and you can access it. It's online in Functional Foods and Health and Disease. And just briefly, these are some of the findings that we found is that patients with more ALCAT reactivity or a greater number of food sensitivities had a lower redox status. Again, ALCAT measures cellular responses that partially, amongst other things, induce oxidative stress. So that wasn't terribly surprising to us. Alcohol consumption showed that there was a higher rate of oxidative stress amongst those people who consumed more alcohol, less resistance, a lower redox status. Alcohol and smoking both, fairly significant. Those with a good diet, and we asked people to self-assess the number of days in a week that they ate prepared foods versus whole foods. And so it's not a perfect assessment. It's not like you can control it as you can a mouse diet, but we did see that those with a better diet had overall a healthier redox status. And overall, we saw people with a good diet had a lower number of micronutrient deficiencies. GI also played a role. We saw that redox status was slightly better amongst people not suffering from GI issues. And Amy, I'll leave it to you.
So this is just the cover of the booklet that comes along with the CNA, Understanding Your Cellular Nutrition Assays. We also have a tutorial that goes along with it, looking at the test results, how to make sense of the test results. And the other thing to keep in mind is, just like every test that's offered by Cell Science Systems, it comes with an opportunity to have a half-hour educational session, complimentary educational session with someone on my staff or myself. And your patients, always welcome to have this as often as you like for every single test that Cell Science Systems offers. If you would prefer that we do not consult with your patients for this half-hour session, we will direct them back to you, and we keep your preferences on file for that, but we're also happy to do that for you if you'd like. And we do not give any clinical guidance, just so that you know, at that particular session. We can't do any of that because we don't know anything about your patient other than the results that we have sitting in front of us. So feel confident in that. And again, if you'd prefer not, we can refer them, of course, back to you, or we can meet with you if you'd like. So if you're interested, if you do not already have an account with Cell Science Systems, you could contact the customer service team at Cell Science Systems. If you're interested in just going right ahead and starting an account, if you haven't already done so, you would go to connect.cellsciencesystems.com. So we're always here to help, though, so please do not hesitate to let us know what questions you have. We look forward to working with you.
This concludes the webinar. If you have submitted questions that were not addressed within this session, we will be emailing you separately. Any additional questions, please email hello@previmedica.com. Thank you for joining us. We look forward to working with you to help your patients promote optimal health.